This talk is about how we can greatly expand our view of studying collective behaviour by considering development and evolution, major areas of research in biology that have been relatively neglected in the study of collective animal behaviour. Collective behaviour is widespread across varied taxa, and some species heavily rely on collective behaviour for safety against predators, finding food, and for solving cognitively demanding tasks like how to steal chicken nuggets. Speaking very broadly, we tend to emphasise one of two areas in the study of collective behaviour, or rather studies fall along a spectrum from collective motion to group decision making. Collective motion is about how local inter-individual interactions allow individuals to come together to form groups, maintain those groups, and how they move together through space and generate different patterns at the group level. Group decision making is concerned with what decisions are made, by whom, when, and how. So you can think of group decision making being more about the large-scale external outcome, while collective motion is more internal, i.e. within the group. However, these are really two ends of a spectrum where the large-scale decision made by the group is generated by the local inter-individual interactions. Typically, even at this scale, we see variation in the time taken. Inter-individual interactions happen quickly, while group-level responses that are based on these, often many repeats of these interactions, take longer. The Nobel Prize winner Nico Tinbergen proposed that to fully understand a behaviour, there are four aspects that need to be understood. We've done a great job in understanding the mechanisms driving collective behaviour, from understanding inter-individual interactions to how groups make collective decisions, such as this figure in the top left of quorum decision making. There's also a growing body of research that directly tests how these collective behaviours are adaptive. For example, a predator attacking a group of birds, as depicted in this figure on the bottom left. However, the historical aspect of this behaviour has been relatively neglected. There are relatively few papers that explore how collective behaviour changes over the lifetime of an individual or colony, or how collective behaviour changes over the evolutionary time. Thus, according to Tinbergen's approach, our understanding of collective behaviour remains only partial. Some of the inspiration for my interest in this area was this review by Dora Byro and colleagues. They argued that longer timescales than we typically study in collective behaviour can be important, but they focused almost exclusively on learning, which is still quite a short-term process compared to ontogeny or evolutionary history. So this led myself and Kate Laskowski to propose this subject as a Royal Society meeting, which took place in May 2022 and was really interesting and a lot of fun to be part of. As part of these meetings, we needed to edit a special issue of Philosophical Transactions. And if you're interested in this topic of collective behaviour over multiple timescales, I'd recommend our review paper that acts as an introduction to this issue. To briefly summarise, what we emphasise in the review is how collective behaviour can be studied at multiple timescales, from the very short of less than a second, such as information transfer occurring between individuals within a group, to longer timescales like the decision the group ultimately makes. And then how collective behaviour varies over the lifespan of individuals and even between generations over evolutionary time. And just in case you're wondering, we cover a diverse range of animals in the review, not just fish. If anything, the diverse range of collective behaviours seen in social insects makes them even more interesting for studying collective behaviour over a wide range of timescales. Some mechanisms of inter-individual interactions between colony members occur in fractions of a second, such as pheromone deposition. Nest building and self-assembly take longer than most collective behaviours seen in fish and birds. In terms of ontogeny, the lifespan of the colony will often be the most relevant, while collective behaviour should be a major player in the evolutionary history of eusocial insects, as they are so dependent on collective behaviour. Here is just one simple example to illustrate the importance of time in collective behaviour. This was a study led by my former postdoc, Hannah. We repeatedly tested the same groups of 8 sticklebacks over multiple days. And we found that over multiple days, as other studies have shown, that collective behaviour diminishes. Groups become less polarised, slower, less cohesive, and less coordinated in their speed. However, we found that the groups also became less similar to one another. This is shown by the plots on the bottom row here, that the intergroup variation increases over time. These changes over time illustrate how a species' average collective behaviour might be difficult to document, and even the variability between groups can vary with timescale. So here are some of the potential questions we could be asking regarding ontogeny and collective behaviour. How does collective behaviour change as individuals or colonies get older? And do these changes correlate with, or could they even be explained by, changes in the sensory systems or morphology of individuals as they develop? And are these changes adaptive? So do they represent shifting trade-offs as animals age? For example, as individuals become more or less vulnerable to predation as they get older. And these are some of the questions we could ask in studying the evolutionary history of collective behaviour. To what extent have collective behaviours evolved independently, or have they been inherited from a common ancestor? To what extent are collective behaviours constrained by evolutionary history, or do they adapt readily as selection pressures change? And how do collective behaviours correlate and co-evolve with other traits, for example in the sensory system or morphology, after controlling for phylogeny? It's worth stressing that as a research community, we've developed a lot of clever tools and approaches that can be used to explore collective behaviour over long timescales. This is really a ripe area for future research. But a major problem we have, which is shared with developmental or evolutionary history studies of other types of behaviour, is that collective behaviour adjusts to the environment and is context dependent in other ways too. So we can test groups of individuals at different life stages or groups of different species in the lab under standardised conditions, but their response to standardised conditions might vary between species. So what we document as a difference between life stages or species in collective behaviour is actually due to a different response to the lab. If we instead test different life stages or species under natural conditions, or mimic each of their natural environments, then we can't tell if the differences in behaviour are due to the environmental variation or the variation due to life stage or the species differences. For contrast, such a problem is not an issue for less plastic traits such as morphology. Here in this example, the pattern and coloration of this frog is not very sensitive to the conditions in which it was photographed. Thanks everyone for listening.